Hi, and welcome to Part 1B of the Kidneys, Chapter 19, where we're going to talk about glomerular filtration rate today. And given that it was preempted by the coronavirus outbreak, it was unfortunate because kidneys have so much to do with whether we live or die. It's actually a little bit frightening in that respect. Um, we talked about these things last time when we talked about the functions of the kidneys. The only thing you don't see on here are hormones, but that's under the direction of producing red blood cells and regulating blood pressure, maybe a little bit of minerals as well. So these functions right here, 6 different ways the kidneys keep you healthy. 8 problems that kidney disease can cause: nerve damage, cardiovascular disease, heart attack, stroke, anemia, high blood pressure, weak bones. It just goes on and on. Things that drive kidney function issues. My grandmother, my maternal grandmother, uh, suffered shrapnel damage when she was in her teens from a homemade firework bomb that went off, and it actually damaged one of her kidneys, and she actually lived most of her life with high blood pressure and frankly died prematurely in her mid 70s, which now doesn't seem like that's incredibly old anymore. So it does have a major role in health, and it's not appreciated enough. Some symptoms, things that as therapists and as caregivers you might be paying attention to, and things that we can test for. And GFR is one of the very big ones that we can test for. Generally non-invasive, and not too difficult to do if you suspect some issues with kidney function. Now, remember from last time we talked about the 3-layer system that we have in the glomerulus. The endothelial cells, which are fenestrated, not only the spaces in between endothelial cells, but actually through endothelial cells, these pores, as well as the basal lamina, and then lastly the podocyte foot processes. These 3 things work together to work on what we call filtration coefficient, which is part of GFR. The other part of GFR is driven by pressure dynamics that we're going to talk about over the next few slides. But GFR, what you do is you're comparing the normal filtration rate, let's say 100, which is 100% of a normal kidney function, and you can ascertain that percentage actually fairly simply. And I'll talk about that later on in this chapter. But if you have reductions in that, we define kidney disease as anything below 60% of normal GFR, all the way down to 15%. If we're below 15%, then we're in kidney failure, and you either need to get a kidney transplant or you have to go on dialysis in order to sustain life. But that's what GFR is. It's the glomerular filtration rate. As mentioned in the previous slide, we have that 3-layer system that drives what we call the coefficient of filtration through here, depending on how tight or how loose, how dense or scarified that membrane is in here. And I'm just going to fill it in here with a little bit of green. That can change very slowly or relatively slowly. It can change over time, and there are age-dependent changes and disease-dependent changes. If you have kidney failure as a consequence of sepsis, that's a function of filtration coefficient, because antibodies get stuck in that membrane and lead to blockage, and then you can't filter anymore. But what we're going to talk about now is what normally drives filtration, and that's pressure gradient. And if you remember in our artery or our capillary filtration reabsorption situation where we had fluid, we had hydrostatic pressure, we had colloid osmotic pressure, and now we're going to add one more. And the one more isn't terribly important if not for the fact that occasionally we get occlusions downstream from the kidney. We sometimes call those kidney stones. You can also have a condition called hydroureter, where you may have some sort of an occlusion that develops, or a malformation that leads to increased fluid pressure. So we have hydrostatic pressure just like before. We have colloid osmotic pressure just like before. And now we're going to add one more, and that's called fluid pressure, that we find in Bowman's capsule past that membrane. So hydrostatic pressure is going to push us out. That's arterial pressure. Colloid osmotic pressure is going to want to pull back in, and fluid pressure is also going to want to push back in. So you have two forces opposing hydrostatic pressure now. So I'm not going to worry too much about the numbers here. I'm not going to worry too much about the numbers. The key is to recognize what is what. So we have P sub H, that's hydrostatic pressure. That's shown right here in this arrow. Okay. We have colloid osmotic pressure. That's going to be pulling back in to into the glomerular, opposing hydrostatic pressure. And then we have P fluid, which again will also oppose. But our net, if we have 55 minus 30 minus 15, so 55 minus 30 would give us 25. 25 minus 15 would give us a net of 10 millimeters of mercury of fluid pressure coming and heading out of Bowman's capsule. Again, if we have a kidney stone, or if we have some sort of an occlusion that leads to a condition called hydroureter, that fluid pressure right here is going to rise precipitously, and it doesn't have to rise all that much. It could rise just a little bit, and it would completely shut down glomerular filtration. So let's move on now and talk about other aspects of GFR. So GFR is mostly driven by net filtration pressure, like we showed you in that figure before. P sub H minus pi, that's the oncotic pressure, minus the P fluid, gives us a normal level of 10, and that's what happens. Now I'm going to show you some mechanisms to prevent major changes in hydrostatic pressure if for some reason your mean arterial pressure rises. For instance, we don't want to pee out just because we get up and start running and our mean arterial pressure rises. That would lead to unacceptable fluid loss. Just getting up out of your chair would cause a major change in hydrostatic pressure. We want to keep this this net pressure somewhere around 10, and we're going to show you those mechanisms here in a little bit. And just a little bit more about filtration coefficient. The surface area is important. That's basically Fick's law. So the more surface area you have, the more ability to filter you have. And then that permeability of the three layers. So a combination of surface area and overall permeability influence the filtration coefficient. Generally, filtration coefficient doesn't change all that much moment to moment. It can change over a period of days or weeks, or if you have injury. The big one that I think about is if you have some sort of antigen antibody complex that shows up in the bloodstream, and I think sepsis when I think that. You have bacteria, and then you have antibodies attacking those bacteria, and they clump up in the glomerulus, and so surface area drops very, very quickly because it's like leaves in a gutter. Once you have those antigen antibody complexes, they either occlude the filter altogether, or they'll induce inflammation in the glomerulus, also shutting off filtration. So it's generally a slow change, but it does change. The one that changes most often is the net filtration pressure. And just a little bit of evidence, and this actually plays into emergency medicine a little bit. Notice that we can have a normal GFR, in this case it's liters per day, 180, but we resorb 99% of that, that we can have a normal GFR even if our mean arterial pressure is somewhere around 80, which is really pretty low, or 180, which is like you're afraid they're going to pop a cork or something. They're going to stroke out on you right on the spot. So a huge difference. over 100 millimeters of mercury difference, and we can sustain GFR. Just for argument's sake, here's normal blood pressure. So we can have significant elevations in blood pressure without altering GFR. Pretty extraordinary, and we're going to talk about how the kidney does that. How does the kidney manage such a wide spread of mean arterial pressure, but keeping GFR very, very stable? By the way, this is one of the alarm bells that goes off if you're taking blood pressures, in an emergency situation, and you see them starting to drop below this. This is when they're going into shock or there's something else. Their kidneys can fail. So one of the things you do is you start taking active measures in order to bring blood pressure up. Or the opposite, if they're up here, they're already hypertensive and they're in extremis, we're going to do something to try to control that blood pressure again so that we don't have kidney damage. So how we can keep that GFR constant despite major changes in cardiac output and mean arterial pressure is by varying the resistance either here or here and here. And we actually have two different mechanisms. One is what we would call a fast mechanism, is known as myogenic autoregulation. And I know that sounds like a mouthful, but it makes sense. It autoregulates. That means that the smooth muscle that's right here does this on its own. It can sense changes in pressure, and it's occurring in the smooth muscle. Myogenic means the muscle or originates from the muscle, and that's smooth muscle here. Autoregulation. Now, what is happening right here? Well, when we have cardiac output that's causing pulse waves, and remember that the renal artery is coming right off of the dorsal aorta. I mean, it is close to the heart, so it's a high pressure system, and the kidneys receive fully 20% of cardiac output. So that's a lot of fluid coming in there. In any case, these smooth muscles can sense a systolic wave, can sense that rise, that transient rise in mean arterial pressure associated with ventricular systole. They will sense that, and they'll actually squeeze down a little bit during that time. Rather than relaxing like other arterial smooth muscle will, these arteries actually contract during ventricular systole. And what they do is by contracting a little bit here, they prevent that pressure wave from going into the glomerulus and causing damage. This is very, very sensitive and almost irreplaceable, or is irreplaceable. But the only way we can recover from that is the glomeruli in the rest of the kidney or the other kidney take up the slack. You can actually survive without a whole kidney, but what happens is that the other kidney actually becomes hypertrophic in order to take up that filtration slack. So this guy contracts during that systolic wave in a process called myogenic autoregulation. We also have a slow mechanism, and this one allows for compensation for longer term changes in mean arterial pressure. Let's say you're under a lot of stress, or you're dehydrated, or other possibilities, even immobilized for a period of time, move to a high altitude or something along those lines. This is a slow mechanism. Usually takes hours or days to kind of make up the difference in the change in GFR. This one's a mouthful also, and it works not only here at the afferent arteriole, it actually also works at the efferent arteriole. And this is what I showed you in that video the other day, where you have the opposite contracting and relaxing. One hand opens, the other hand closes, and vice versa. This slow mechanism is called tubulo-glomerular feedback. So the tubules, the filtrate in the tubules feed back and cause changes in smooth muscle tone at the glomerulus in order to vary the hydrostatic pressure in the glomerulus. And we're going to actually break that mechanism down quite a bit. So myogenic autoregulation and tubuloglomerular feedback can both work here at the afferent arteriole. Only tubuloglomerular feedback works at the efferent arteriole. So if we have a systolic wave, a systolic bolus of blood coming through here, the myogenic autoregulation will kick in and will squeeze for just a moment in order to lower that incoming pressure, keep hydrostatic pressure so that we have a stable GFR. If we're talking about tubuloglomerular feedback, you have a longer duration constriction, and that can then reduce GFR overall. What we would also have with tubuloglomerular feedback is that you would have a dilation on the other side. So they kind of work hand in hand, they work in tandem, whereas myogenic autoregulation only works at the afferent arteriole. And so I just have some examples here. This one shows the efferent arteriole in this one, but this is the key figure question. If you can get this question down right here, you should have a pretty good handle on what's going on in terms of the response, okay? The constriction dilation event here at the afferent or efferent arteriole is the response that leads to changes in GFR. We're going to work on the stimulus and the mechanism for tubuloglomerular feedback. So the ways that we can regulate or alter, the word is modulate, glomerular filtration rate include that myogenic autoregulation response, and this is from the vascular smooth muscle, and it senses that systolic pressure wave coming down the vessel and is able to squeeze in time to prevent that elevated pressure from going into the delicate parts of the glomerulus. It is very similar, in fact, almost identical to autoregulation in other arterioles. And the one I'm thinking of is the coronary arteries. Coronary arteries actually constrict during cardiac systole, during ventricular systole, to prevent that pressure wave coming straight off of the aorta. I mean, it is those things. Those are the very first vessels that branch off the aorta are the coronary arteries, and so they have to constrict in order to dial down that huge amount of pressure, relatively speaking, coming off the aorta. So we see this more than just in the kidney. We also see it in the coronary arteries. We have the slow mechanism, tubuloglomerular feedback, and this is going to also use the vascular smooth muscle, but it's going to use a paracrine control system that we're going to dial into much more specifically here in a few minutes. And then we have one more. I guess I sort of led you on saying there were two ways. There's actually three: hormones, and yes, there is a little bit of neuronal influence that can change the arterial diameter. With hormones, you can alter the filtration coefficient of the filter itself. Again, this is very, very slow. So we have the fast here in myogenic autoregulation. We have the slow tubuloglomerular feedback, hours or days later, usually 2 or 3 days. And then the very slow, which can take, if you're using the neurons, that may be a little bit faster, but we're not going to go there. We're not going to worry about that. What I mean is with the filtration coefficient. In order to fully understand the mechanism that is tubuloglomerular feedback, we need to look at this anatomically. So right here, and I'm going to draw this in blue, we have our glomerulus, and that's where we're filtering. And then what happens, and I'll draw the rest in green, is that filtrate moves into what's called the proximal tubule, descending down the descending loop of Henle, where we lose water to osmosis. And then on the ascending loop of Henle, we are actually exporting solute. And then that tubule runs right in between, and I'm going to draw those in, let's draw those in purple, right in between the afferent arteriole and the efferent arteriole. It runs right through there. And so that proximity is what drives the signaling process or assists the signaling process. So let's look at it zoomed in a little bit here. So if we look over here, this is the ascending loop of Henle coming right up through that V that is created by the afferent and efferent arterioles. As it crosses by there, there's an area known as the juxtaglomerular apparatus. And there's these macula densa cells. I'm not going to worry about that detail. But this juxtaglomerular apparatus includes special flow and osmoreceptors in these cells right here. And they can detect the amount of flow as well as the osmolarity of the flow coming up through that ascending loop of Henle. So now let me show it to you in a cartoon form. We're going to walk through this. A lot of students seem to have trouble with this mechanism for reasons I don't fully understand. So I'm going to try to take this very, very slowly, make sure that you understand how these steps work and are able to indicate what's going on and where. So the first thing that happens, and in this case, it could be a GFR decrease, but for our purposes, we're going to say that GFR is increasing for this particular situation. So what's happening is glomerular filtration is increasing. Okay, and that generally occurs because what's happening is that you have some increase in mean arterial pressure, some increase in hydrostatic pressure. So when we have that increased GFR, that means that there is going to be more fluid moving through, more filtrate moving through this tubule. There's going to be more of it. It's going to be like kind of like a cave passage that's when there's a rainstorm outside, we see more water flowing through. In this case, it would be more filtrate flowing through this tubule. Well, we haven't talked about it yet, but when we get to around, when we get into this proximal tubule, if you will, when we get into this proximal tubule, we have a lot of enzymatic type carrier situations going on. That is to say that there are specific carrier proteins secreting and reabsorbing material through here. Let me give you sort of a lousy analogy. Let's say you go to a cafeteria and you're in the cafeteria, but instead of the line moving on at a very leisurely pace, where you're moving only a foot or two every few seconds, and you get a good long look, especially at those desserts, right? You got a chance to choose things as you move slowly. Instead, let's say the cafeteria is just throwing people through. They're just flying through there, and you only have 20 seconds to get through that cafeteria line because the line is moving so fast. Do you think you could effectively interact with the displays and the people that are serving up the entrees? Of course not. By the time you get through that line in 20 seconds, you'd have hardly anything on your tray. That's the same sort of story that's going on in the proximal tubule. Because these are enzymatic exchanges that are going on, rate does play a factor. If this flows by here, you won't have as much time to extract material, and that's a lot of what's going on. 2/3 of the material that is returned to the bloodstream occurs here in the proximal tubule. And then, of course, we flow down through the loop of Henle and up through the ascending loop of Henle. And again, we're going to have more flow through here, and it's going to be less concentrated, less time for equilibration all the way through this process. So once we get here at 3, the flow past that macula densa, if you will, that sensory region of the juxtaglomerular apparatus, right here inside this notch, it's going to see more flow, and it's going to be less osmolar. It's going to have more water in it. Well, those macula densa cells will sense that flow in particular and sense the osmolarity and go, hey, we need to slow down GFR. And so they'll send paracrine signals both to the 5 here, and I'm going to draw in another one here. I'm going to draw it in black. And it's also going to send a signal up here to the efferent arteriole in order for it to open up. So it will tell, in this case, we want to slow filtration down. So we're going to close this one and we're going to open this one. That will lower the pressure in here. That afferent arteriole constricts, and we could say the efferent arteriole dilates. Will lead to a change in the pressure because we have changed the resistance. By increasing the resistance here, we have changed the pressure in here, which means less flow. Less hydrostatic pressure in the glomerulus means less GFR. So to restate this, what we are looking at here is a negative feedback response system, but instead of a silver thornogram, we have it shown anatomically. So if we have too much pressure, we have too much filtration. Too much filtration means too much flow, and the osmolarity is too low. The macula densa will sense that and send paracrine signals to the afferent arteriole to clamp down and the efferent arteriole to open up. That causes a reduction in pressure due to the resistance therein. So we have a reduction in pressure and a reduction of flow. And then vice versa. Too little flow, we have too slow flow at 4. They send a signal out for the 5 to open up, the afferent arteriole, and for the efferent to close. That causes a rise in pressure here, and we restore flow back to its homeostatic set point. Again, I've always wondered why there does seem to be a population of students who has a really tough time with this mechanism. And as always, just reach out to me. Let me know what's giving you trouble. Try drawing this out. Draw it to know it. Work through it. Macula densa, juxtaglomerular apparatus, afferent and efferent arterioles, mean arterial pressure and filtration. Okay, it should be fairly straightforward. If you practice it a couple of times, you should be able to identify it in any type of multiple choice or short answer question. And so that concludes part 1 of the chapter 19. So we have a part A and a part B. So this is the end of part B. Understand that GFR is a major diagnostic tool, especially with folks who are candidates for surgery, folks who are in intensive care, folks who are under significant stress or medical conditions. It determines whether or not somebody gets on a transplant list, somebody gets sent to dialysis. A lot going on here, so it's a very important aspect of physiology that plays an everyday role in clinical practice. We're going to be moving into part 2, where we're going to talk about the tubular side of the equation. That's the secretion and reabsorption. So thank you for your time. And let me know if you have any questions. I'm always glad to help out any way that I can. See you soon.